Hi everybody, welcome back to our next video on cells. This video we're going to start by talking about the two eukaryotic cells, the two types of eukaryotic cells. Remember, prokaryotic cells, those are the cells that have no organelles on the inside. These eukaryotic cells are the more complicated kinds, and we find that they fall into two categories. The animal cell, we see the animal cell here on the right. We can see there's a whole bunch of different little organelle things. These are all the little drawings that we put inside this circle. But a main important factor that you'll be able to identify an animal cell with is the lack of shape. So notice this sort of general, sort of circular, spherical shape. Now there are animal cells that come in long shapes, like your muscle cells. There's ones that are very spherical, or spherical and flat, like your blood cells. There's all sorts of different types, but this is one of the main features we see in animal cells is sort of a lack of very structured shape. Over here we see the plant cell, and the plant cell has a cell wall, and that typically causes the plant cell to have a very structured shape. In fact, this is the reason we use the word cell is that the first scientist to see cells was looking at plant cells, and they thought that these looked like the little rooms that monks worked in. And monks work in little rooms called cells to write scrolls and to copy and to work as like scripts, to work on scripts and things like that, scripture and and before there was a printing press, monks would work in these little cells and they would do like their little writings and they'd live in those cells and things like that. So he thought of them as a cell, same thing as a jail cell, I guess, as well. It's a small room with a repeating structure. And so we do see a bunch of other features here. We're going to talk about each one of these in time. Some of the things that we care about in this class are going to be the mitochondrion, which we find in both of the cell types. So there's another mitochondrion. We might care a little bit about chloroplasts. These are unique to the plant cell and also to another type of cell we'll talk about later on. And that's about it. Then the nucleus is the other important factor. It's the factor that's in both of them. It's a feature of eukaryotes that they have a nucleus. And then we also have the ribosomes, which we also noticed in the previous example of the, you'll notice that ribosomes are also indicated in our prokaryotic cells. So those are all three or four, various features that are very important to the structure and function of the cell. All right. Now we have a cell. Some people would describe the cell in a way that is similar to a factory. Uh, and so let's look at this factory first and see if we can identify some of the ideas here. So in the factory we have a a central computer here, number one. And that central computer, this controller person, is telling all the factory workers what to do. We also have someone who is storing blueprints, right? And they have all these different blueprints on all the different things that they can make, and they're going to send those blueprints around the factory to tell them what to do. We also notice that this factory controller, so maybe the manager of the factory, they work inside a little bubble. They have their own little bubble here. Now, they send little signals and messages to the other parts of the cell. One part of the cell is a little factory that produces energy, and the controller can tell that factory to produce more or less energy. They can also send messages over to this person here, who's actually taking the blueprints from right here. He's taking those blueprints and he's using them to make something. He's making something we're calling a protein here, and he's making these proteins. They're kind of just landing on the floor. They're not doing anything useful. The proteins are eventually getting over to this person, and they're sort of packaging whatever that stuff is that's coming out of the maker space here. And then they're also getting transported around the factory by number 7, including in and out. And finally, there is another section over here where there are raw sugar kind of being packaged up into sugar, raw ingredients being converted into sugar for use later. So let's see. Now I think some of my ideas that I have originally labeled over here might be actually a little bit different. So number one, the person in charge. Well, that is definitely the nucleus. So that's what I've put here is the nucleus. So we can kind of think this person is supposed to be like the nucleus. They're the boss. So the boss. And by the boss of a cell, that means that they are in charge of producing all of the instructions that the rest of the cell has to follow. Each piece of the cell can do its own particular things, but it is the boss who really is going to is going to allow them to do those things or encourage them to do such things. The things that they're going to be doing are going to be based on instructions in the chromosomes. So these are the instructions or the blueprints. They're all the blueprints and instructions are stored in the master set inside the chromosome. They will eventually send copies over to number 4 here, but they are the original master copies, master blueprints. The master blueprints. I guess I should call this, but we're giving it the cell name. Okay. So this is the chromosome. And notice that a boss only knows what they're able to do if they read the blueprints, right? They can't do anything that their store or their factory doesn't have instructions for. So the nucleus is largely, indebted to the chromosomes for what it can and cannot do. And then we have this box here. I'm going to just point this box out. This is what we're going to call the nuclear membrane. Nuclear membrane. And I'd encourage you to come back to this image after you've kind of worked your way around later. So the mitochondria, this is the powerhouse. This is where the power for the rest of the cell comes from. Powerhouse. And then we have the ribosomes over in number 4. So that's really the ribosome. It takes the instructions and it produces the product. So this is the factory production. So they're the ones who are in charge of producing, making sure that something is coming out. They're producing protein, and that protein gets packaged up. And I'm going to actually say that this is not a vacuole, I don't think. I think that's the wrong image. I'm going to say that this is, we're going to put the endoplasmic reticulum down here. So this is the endoplasmic reticulum. And that's the packaging. And packaging. So they take the raw protein and they kind of fold it up and make it into nice little packages that can be sent out. And then to distribute it, number 7 here, this is going to be the Golgi apparatus. And now hopefully I'm not hopefully I'm not going to regret any of those decisions. Okay, so now I have number 5. Now, number 5 originally I was thinking, oh, maybe that's supposed to be a chloroplast. It's making sugar. So certainly cells make sugar, like plant cells make sugar. So it could be that, or it might be if we want to think about animal cells, which we are thinking about animal cells today, this is probably a lysosome. And it break down raw fuel for consumption. Breaks down raw fuel. Okay, I'm going to have to make that one text because my typing is really bad there. Okay, lysosome breaks down that's probably good enough. Hopefully you can read that at home. That's a little too big. Okay. I know. My videos could probably use a little bit of editing, but, who's got time for that? Okay. Let's call it packaging department. Okay. And we're going to cross out the vacuoles again. All right, and then we do have the cell membrane, which is the last step, which is 8, coming in and out, prevents, the walls of factory. All right, so we're going to say this is lysosome, and this is mitochondria. this one is our ribosome, and this is our ER. So one of the things, I mean, I know this doesn't make a lot of sense right now because we haven't talked about each of these pieces, but we'll come back to, hopefully you can come back to this image and it's going to make a lot more sense afterwards. But the idea is that we're moving things around. Each of these organelles has its own function. So organelles, they make it possible for different chemical processes to take place at the same time. They have unique structure and function. We have protein synthesis as a function, energy transformation, digestion of food, excretion of waste, and cell division. All of these have to happen. It's like a little tiny person, right? Just like the human body has different organs for different functions, so does the cell have different organelles for different functions. So first we have the nucleus. This is our sort of data center. That's going to direct all the cell's activities. It contains your DNA. It stores information that you need to make proteins for cell growth and function and reproduction. It's surrounded by a nuclear envelope, and it has pores that allow substances in and out of the nucleus. Inside, we have one feature I want to point out, which is that I don't have notes on otherwise, which is the nucleolus. And this is where ribosomes are made. Later on in this course, we're going to do a whole section about the types of material we find in the nucleus, chromosomes and DNA and RNA, and we'll talk about how that's an important feature there. So I need you to kind of keep that in mind for a little bit later. Now, interestingly, if you look right here, there's sort of like a connecting path here, and that connects out into these folded sacs that surround it, and those are the endoplasmic reticulum, or the ER. Okay, and we're going to talk about that in 1 second when I scroll down here. So here's the endoplasmic reticulum. It has two pieces. It has a rough endoplasmic reticulum and a smooth one. It is a membrane of folded sacs, and they're all in a system together, and it's the site where protein and lipids are formed in the cell. We are really focused on proteins in this class. We're not going to talk about lipids at all, really. But proteins are produced by the ribosomes, which are attached to the endoplasmic reticulum. They're not attached. They can be free floating as well, but they're adjacent to the endoplasmic reticulum. Some of them are attached, particularly in the rough endoplasmic reticulum, and those ribosomes help produce raw protein. But then when those raw proteins travel through the endoplasmic reticulum, that raw protein gets converted into the finalized packaged protein that we want to send out into the world. So it is important for folding. We're going to call this, so the ER is essential to proper folding of proteins. The final step of protein synthesis. So folding a protein is something we'll discuss in a lot of detail in our next in a following unit. And if without proper folding of a protein, the protein actually will not function. So we need this endoplasmic reticulum to fold the proteins. It makes those proteins function, and so without this fold, the proteins won't function. Unfolded proteins will not function. Okay. The ribosomes are the site where protein is formed. So they are mostly the ones in charge of protein formation. They're the little protein factories, and they're located in the cytoplasm freely, or they can be on the endoplasmic reticulum, where they can insert their unfolded proteins directly for folding. The Golgi apparatus is kind of like a mail room. It's a flattened sac again, which is always good, but it has the ability to sort and package these proteins, these folded proteins, into little sacs, and these sacs are called vesicles, and the vesicles move around the cell. So maybe in our previous little image that we talked about at the beginning, it could be that little guy on the cart. He's kind of the vesicles, but if we think of the vesicles and the Golgi all being part of one thing, then it's the one that's kind of traveling around. Vacuoles are places where there are temporary storage of materials. We have very few in an animal cell. The animal cells use and interact with chemicals in a different way, but vacuoles in plant cells are very important for storing water, as water is part of the photosynthesis process. So we need lots and lots of water to keep those cells functioning. Lysosomes are similar in appearance to the other vesicles we've discussed. So the vacuoles is a storage vesicle. So it's a you know vacuoles are a type of storage vesicle, and whereas lysosomes are a type of disposal vesicle. Are disposal vesicles. So if you think about it that way, that the lysosomes are kind of the opposite of a vacuole. A vacuole stores stuff for use later, and a lysosome disposes of things for disposal later. So you can fuse with other vacuoles to digest the waste inside of a vacuole. They are filled with all sorts of enzymes. So they're filled with enzymes. I notice it's not in here, so let's call filled with enzymes that digest waste. So that's their main thing. Now, an enzyme is a type of protein that's important to making sure a chemical reaction happens, like destruction of a waste product. Too much waste product in your cell, bad news. It's going to poison itself. Centrioles, we don't care too much about centrioles. They're found in animal cells. They are the only found in animal cells. So if you want to pick something that's going to make an animal and a plant cell different, this is one of those. They are made of microtubules. Microtubules are long, hollow cylinders that form a skeleton inside of the cell that help the cell from collapsing. Even if the cell loses a lot of water and dries out, it might not completely collapse because of this cytoskeleton. And these microtubules are really important to that, but also these microtubules can expand and contract, which is really important for moving things around the cell during mitosis. So they only have a function during mitosis and meiosis. So that's only during cell division. We're going to talk about cell division very shortly. Mitochondria, these are the powerhouse. They are really interesting. They have their own RNA data. So like this is a type of chemical that stores information. We call it has a specific name. It's called mRNA, and I believe we've already kind of mentioned mRNA in another class or in some videos about phylogenetics and how they establish phylogenetics using mRNA. That's what we're talking about. So this is another reason we want to walk through our cell, is that the mitochondria has its own RNA, and that RNA indicates that the mitochondria may have been its own creature at some point, its own bacteria-like creature. Chloroplasts have a similar case. They, I believe, even have their own RNA as well, and they also have similar structure to the mitochondria, but they are specialized for photosynthesis. So these are going to be these are only in plants. Cell walls we find in plants. They are made of cellulose in a plant. So mostly made of cellulose. So I'm going to type that out. Mostly made of cellulose. in plants, and chitin in fungi. And so fungi have a similar sort of cell wall. I believe the cell wall is different in structure and function a little bit, but it is similar enough that we can say that they are somewhat found in fungi as well. Maybe some only specialized cells of the fungi. So then we also have some other features that we specifically find in a lot of different cells. Cilia are found on lots of different types of cells, like including some of your own cells have some cilia in them that help them swim around. They are these short, numerous hairs, sort of projections. They're not really hairs because they're so much smaller than that, but they are useful for communication in bacteria. So bacteria can use cilia to communicate and to share DNA with each other. Uh, so cilia are used for individual or for sharing. Let's just go sharing. Sharing sharing genetic information in bacteria. This is called horizontal gene transfer. I'm going to introduce that here, but we won't come back to it for a little while. But that's one of those concepts. And then flagella are usually used for motion. So cilia typically are used to kind of stick to things. Um, so also used uh, to adhere to surfaces. Adhere to surfaces. So they can be used to kind of stick to things. Flagella are used for motion. So flagella are used for moving the cell around, especially in unicellular creatures. Also in sex cells. So lots of different creatures have flagella in their sex cells. So human beings, of course, the sperm has a flagella, but we also see flagella on spores. I believe even fungi have flagella spores, but even some types of ferns and a bunch of other organisms like that there are plants that can send flagella-bearing sex cells through the water supply so that they can mate with adjacent plants. And same with fungi. There are examples all throughout the kingdoms of this kind of action. All right, hopefully you found that enlightening and interesting. If not, we're almost done cells anyway, so we'll get back into something else in the next video. Please try to get a look at this practice. If you need to, you might have to go and do a little bit of research. Um, and then we will come back and talk about the kingdoms of life and some of the things that we learned there. All right, thanks everybody. Have a great day.